been here in probably about 15 years. It's lovely to be back. I'd like to tell you a little bit about work we've been doing. Actually, some of it really goes back almost 30 years. When I spent a couple of years in industry, in the farm industry at Merck and Company, which is really what turned me from a mainstream chemical engineer into something like a physical chemist. And what we do kinetics, but basically we try to understand catalytic reactions. We think of it as a puzzle, putting pieces together. And there's lots of different ways we can do that. We heard already this morning about some. You can use computation, you can look at crystal structures, you can do spectroscopic analysis to look for intermediates. We do all those things, but we also start with kinetics. We start with what I call global kinetic analysis. We're not trying to use kinetics directly to see an intermediate space. We're trying to look at the form of the curve and then build a mechanism around that. My take-home message is do the kinetics first. If you do this first, it'll tell you where to go next. Because you can spend a lot of time looking for a reaction intermediate via spectroscopy or trying to crystallize it. If kinetics tells you that it's after the rate limiting step, profile, like what I just showed you, really goes back more than 100 years to the Michaelis-Menten equation, which was the first description of a catalytic reaction. It was an enzyme. But enzyme reactions are the same as basically you have binding to a substrate, a catalyst to a substrate followed by a reaction. That works for chemocatalysis. It works for heterogeneous, homogeneous enzyme catalysis. And I don't know whether the younger people, some of this is going to be kind of basic background because we're so broad here. Heterogeneous, homogeneous, people doing a lot of different things. So I apologize if some of it is too basic. But the young people may not know the Michaelis-Menten equation, that Menten was a woman, Miss Maud Menten, in 1913 in Germany, working as a scientist. I worked in Germany in the 1990s, and I can only imagine what it was like for her in 1990s. So this was the first time that people were able to describe a catalytic system. But you can see that the rate law is actually complex because you have substrate in the numerator, multiplicative in the denominator as an additive. So it's not a linear. You don't get a linear curve. And so in 1934, the first method that was published to linearize the data basically just took that equation and turned it upside down. 1 over substrate. And now you have a straight line with a slope and an intercept that allows you to extract the meaningful parameters. Km, which is a Michaelis constant, is like an inverse binding constant in our terminology. And that was incredibly enabling. It actually was enabling for generations of scientists. For the first 125 years of Journal of American Chemical Society, 2003, they went back and looked at all the citations. The Langrenberg paper was the most highly cited paper. It was cited once every 2 days since 1934. And so, because it was empowering, people were actually able to get information that was hard to get out of this equation, but they could get it out of this when you linearize it and turn it upside down. We also, as I'm a chemical engineer, and we talk about power law. We actually also try to relate this expression, Michaelis-Menten, to a reaction order. So we'll talk more about that. Chemical engineers will call it the form we use 1+ equations. So we were inspired by this to think, we need new ways to extract information from our data. We've been doing the same thing for more than 100 years. And what people do now, we've got all these powerful tools, React IR, that Google has shown here. We've got lots of in situ methods to get data points every 2 seconds or something. What we do often is we take an initial rate, a delta C, delta T, and then we throw away all these data, and then we start over at a different concentration and do another delta C. Because this is not a straight line, and sorry to say that synthetic organic chemists really don't like when 2 variables are changing at once, so they don't like to look at things like this. But we know that there's information in that. And so I basically developed what we call reaction progress kinetic analysis to try to make that easier to extract the information from those profiles. I'm not going to go into the whole methodology. It's very simple, but you can read the review papers if you want. But basically, what we get out of reaction photokinetic analysis by looking at the full profile is we try to get reaction orders. We get these values X, and then we try to relate it to the catalytic expression. So the order X is sort of a shorthand for trying to understand the binding constant, how strongly a substrate binds. This is the simplest case of a Michaelis Menten. I mean, most of our reactions are more complicated than just A going to B. That's a decomposition or an isomerization. We're usually trying to put two molecules together. But the same idea works for more complex cases. And the idea is, if you look at these three graphs here, basically there's a fit, a power law. It's just a fit, but catalytic, the equilibrium constant here and related, that's the line, and then the data points are what would be if I fit it to a power law. Now it only works within the range of concentrations, a certain range, because X is going to keep going up forever, and we know that Michaelis Menten can enter saturate. But within that range, we can use as a sort of proxy for the equilibrium constant. We can use a value of X. If it's a high value of X in this case, then the equilibrium constant is going to be close to 1. If it's very low value of X, then the equilibrium constant will be higher, because it essentially changes the relative amounts of the two additive terms in the denominator. Very simple idea, and it can be a very powerful way to start, even if you don't know a mechanism. If you use programs, there's a lot of programs out there to help you, like Dynachem. It's very popular, especially in industry. You have to write out all the elementary steps. And if you don't know that before you start, trying to figure out X's and Y's orders is a really good way to start trying to develop a mechanism. And so I'll just show you a couple of cases that we've done. Basically, this is a Whipple-Hartwig. We were talking this morning about CC and CN coupling. This is a very famous Whipple-Hartwig CN coupling reaction. We use two different amines, you get very different looking profiles. This is heat flow. I love reaction calorimetry. It's my favorite tool. That you get reaction rate is you get directly. So that's rate on the scale. And you can see that with the blue, which is a linear hexylamine, it's a nice exponential curve. It's first order kinetics. But if you use a hydrazone, you get a zero order kinetics. This is rate. So the rate is not changing until the very end of the reaction. So what does that tell us? If we build a hypothesis for a mechanism where you do oxidative addition followed by addition of the aryl halide, followed by addition of the amine and then reductive elimination. If you first order kinetics, one potential idea is that the resting state is here and the rate limiting step is the oxidative addition. You would get first order in aryl halide and zero order in the amine. If you get overall zero order kinetics, then you think about this being the resting state, because basically, to make the product, you're not changing either of the substrates. So you get zero order kinetics in substrate. And we've shown this by many other, we've actually identified this complex by NMR and mass spec. We don't look for this one because it's fleeting. In both cases, or not either of those two cases, you're not going to see it. So that's a simple case, but often we're getting orders that are non-integer. And they have no chemical meaning themselves. I've had people say, well, I got an order of 0.6. I understand it's 0.5 because that would be a dimerization. It's not. It's an approximation for how far away you are from standard, from what should be in the numerator. So this is my fantastic colleague Jim Kwan Yu published this paper early on in Science. And then we went into, this was what they wrote originally, and then we went and looked at the mechanism by the methods that I don't have time to go into, but by RPKA, we were able to see that zero order in this substrate, negative order in this substrate, negative order in the product coming off, and first order plate. It's like, who's driving this reaction? Nobody's, none of the substrates are driving. But so we could turn those non-integer values into a 1+ equation, and what that told us is that we've got off-cycle species, both of this substrate, olefinic substrate, and olefinic product are present off the cycle. This is not working anymore. Yeah, there it goes. And so that's how we use this idea of using non-integer orders to come up with a proposal for a mechanism, and then you look at that and say, well, what can I do? Can I go look for that resting state? Use my other tools. So one final example of this simple idea is we did aldol kinetic. This is back before anybody had seen an enamine species. It was called the elusive enamine. Everybody knew it should be there in a mechanism like this. When you do nucleophilic addition of a ketone, you have an iminium that collapses to an enamine, you add the aldehyde, add water back in and get the product. You can write out a steady state rate equation for that, and it's horrendous. Nobody wants to try to fit that. And if they do, all the constants are constants. You can't get those numbers for those constants, but we can try to simplify it. So we actually found that the rate was 0.45, close to half order in substrate 1, 0.9, close to 1 in substrate 2, and negative order in water. So what that told us is that we have to have water in the denominator. We've got 1 and 2, both substrates here, but the K for 1 is going to be bigger because it's pulling more 1 further out of the numerator. So basically what that told us, later Ruth Geschwind in Regensburg did fantastic NMR work and found this elusive enamine. But that's essentially the resting state is mainly here, but a little bit here. And the rest, and the rate limiting step is adding the aldehyde. So by starting there, up here with this power law, we were able to look at a plausible proposed mechanism and come up with where we think we could be looking for intermediates. So that's all well and good. We do this all the time for every new system. And most of our systems are fairly simple. A catalytic cycle with a couple of intermediates, first order, partial order in both, zero order. But we're like, most of the systems we're looking at now, newer reactions are much more complex. We've got more than one cycle in almost everything that you're hearing about, photocatalysis, electrochemical reaction, cascade reactions. Anytime you make a radical intermediate, you're probably making it in one cycle and then trying to deliver it to a cycle. So what that means is that we're often doing a reaction where we have a substrate coming from one cycle going into a second cycle. We have no idea what its concentration is. So it's very hard to do kinetics the way I just told you. So what we're trying to do now, basically, if you have a cascade, I think of it like a relay race. And how do the runners know what order to do things in? I know that they seem to. Or these kinds of coupled cycles, they're cogs, and they have to be balanced. If they're not balanced, you could be making this reactive intermediate, and if it's not ready to go, the second cycle's not ready for it, it may kamikaze out, and you're going to end up with low yields. So we're trying to see if we can use the same kind of approach to understand these more complex multi-cycle systems, multi-component multi-cycle systems. So I'll first look at just this. What I'm going to show you now is just modeling. It's not experimental data. I was just trying to use this to show people that we can get like what looks like weird behavior, but what is understandable once we realize that we're dealing with more complex systems. So start with a famous system like this. And that was one that was Dieter Enders published almost 20 years ago now. And it's a cascade where you do a Michael addition with a diaryl ether organocatalyst, and you make this product. And you can stop there, but if you put in cinnamaldehyde or acinnamaldehyde, then the product of that reaction can feed into another cycle, and you can make a more complex product. He basically does a cyclo addition, sets 4 chiral centers at almost perfect E. I mean, it was really amazing. That was the first one. And then there's like, I think we counted 246 papers since then doing similar things. People are saying, by judicious choice of reagents, we have that they're all using the same Michael addition, or maybe a thiamichael, as a Michael, but and they don't know why. I know why, but we can talk about that. But I thought let's see what happens if we just try to do RPK on this system. And I'm going to show you 3 cases. What we're going to look at is it's the same catalyst. This guy is in both cycles, so they have to figure out how to partition the catalyst between the different intermediates. So we've got 2 intermediates in this cycle and 1 in this one. And so let's just look at cases where we have, they're all reversible reactions. So strong binding to give us, strong binding here or here or here. And the rate, if you don't know a mechanism, the rate of the reaction can always be written as the rate of the product determining the product forming step. You can't make product faster than 4 and intermediate 3 get together to make the product. So let's just see what happens to the rate that we observe. This is all just simulations, but let's look first at case 1, strong binding at intermediate 2. So a lot of the catalysts will be sitting here. And what we see is nothing really unusual. Basically, what I'm doing is I just double the concentration of 1, 2, and 3. Standard reaction, double 1, double 2, double 3, all the rates go up. you can see that you start, you change the relative amounts of the intermediates. These are basically the fraction of each species, 5, in 1, in 2, and in 3, at 50% conversion. I think I picked 50, but just to give you a snapshot of what it looks like. Nothing really unusual going on. Positive order kinetics in all 3 substrates. You increase the driving force, you think you get a higher rate. So that seems to make a lot of sense. So let's look at case B, where intermediate 1 is now binding fairly strongly. So what happens now? Well, what happens now, I double the concentration of 1 and the rate goes down on the standard condition. We get negative order, negative 0.45 order in substrate 1. That sounds like it's an off-cycle species. And if you didn't know anything about the mechanism, you'd say that. If you look at it, it's kind of off-cycle. It's off the cycle. It's not in the product forming cycle, but it's definitely on the productive pathway. But this is the kind of behavior you can get. And you can see why. Standard had this much of, this fraction of intermediate 1. We double 1, we have a huge amount here, but at the expense of intermediate 3, because there's only so much catalyst to go around. So basically, intermediate 3 is much lower, and since the rate depends on intermediate 3, you get a lower rate. So I did all this modeling, and then I got my postdoc to redo it all because I wasn't sure I believed it. But it makes sense once you look at the intermediate distribution and along with the kinetics. Now this is great in simulations. In real life, we may not be able to see those intermediates, so we're going to have to try to learn from this to infer what can happen. If we tell most of the catalysts to go here, is that going to hurt this rate here? So case C, strong 3 bind. So a lot of the catalyst is going to be here. That should be great for us, right? That's a driving force in this reaction. Well, we double 3 and the rate goes down from the standard conditions. This is standard. All I did here was double the amount of 3. The rate goes down. Negative half order in 3. Doubling 3 decreases the rate. I should have a lot of this. But remember, there's 2 things in this rate law. There's also 4. And so when you look at looking at the intermediates, we didn't really, doubling 3 didn't really change the amount. We had a lot of 3 for most of these cases anyway. Doubling 1, doubling 3, doubling 2, we went down. But if we just doubled 3, the rates decrease. So we have to look at 4. This is the substrate for this reaction. And we don't know a priori how much is there. So if I look at that, actually, over time, for the whole reaction, standard, the amount of this 4 changes significantly, goes up and then down. Doubling 3 decreases. So that explains why the rate went down, why it looks like a negative order in substrate 3, not because we didn't decrease this intermediate, but we decreased the amount of 4. So this is just the sort of thing that when at first glance it looks non-intuitive if you're just thinking about one cycle and mechanism and kinetics. But once you think about, if you can draw yourself a couple of cycles of what might be going on and see how the catalyst is partitioning itself, your explanation becomes rational. So basically the same idea when we have certainly equilibrium constants. I have another case I didn't show you, but and then we have the orders and substrate for those. And so it's like putting the puzzle together by starting with our power law, and then hopefully we can do this for cases where we don't know the maths. Start from the power law and then work backwards to a mechanism that fits that puzzle. I mean, it's a little bit like playing Cluedo, right? It's like substrate, intermediate 3 in the second cycle with substrate 4. But we like to solve puzzles. So the second example is we also don't yet have the experimental data, time course data on this, but Song Lin has been doing a lot of electrocatalysis. And this is a case also where we're always going to have multiple cycles. And he has 2 anodic events going. And this is the way he draws it. He did, original work, and then he also wrote a review about a bunch of differences. So we just took this one to study. When I'm thinking in terms of systems chemistry, I write it out like this. Basically, there's 2 species that are in both anodic events. There's the first anodic event here, and then there's the second anodic event here. And so, and there's an off-cycle reaction where this intermediate that's formed here gets together with substrate 7, this guy, to make intermediate 11, which then reacts with this copper to make the product. So a lot of things going on here, off and on the cycle. And I haven't even taken into account like a decomposition. These are radical species. So if they are not ready, if 10 isn't ready to react with 7 to make 11, 10 is not going to hang around. Or if 11, if this gets made and this guy this tends to be the recipe, so it's probably okay to go on to the product. But so even without taking into account deactivation issues, you can get weird behavior. So Solomon and his postdoc had a disagreement about what they thought was rate determining. I can't remember who thought which, but either the addition of the p-centered radical to the alkene, which is this one, or rate determining formation of the p-centered radical, which is either formation of 10 or reaction of 10 to make 11. And so can we tell the difference with kinetics between those two mechanistic hypotheses? Well, when we looked at the reaction, it was actually weird. The kinetics look kind of, there's a little bit of induction period kind of looking, but otherwise they don't look that weird. Standard, doubling 9, doubling 7, doubling 8, not much happens. This is a little bit faster, but doubling 7 and 8, the rate jumps. Okay, that's for case 1. And I took out the slides to go into why that's happening because I'm afraid I'm going to run out of time. But so the second possibility where the p-centered rate determining formation of that radical, now we can actually see a difference. Doubling not 7 or 9 give you the same rate as a standard. Doubling 8 gives you the same ratio before it. That didn't change it, but this is the same as if we double 7 and 8. And so 8 is running out, or 7 is running out. Sorry, because we didn't double it. So we can only go to the same conversion as these guys. So it's giving the same rate. So if those are the two possibilities, we just do the kinetics of the reaction. We can tell which of those steps would be rated in an ideal world. That would be the next thing to try. I may actually go on faster than it because I'm actually now at the end. I can go into exercise if anybody wants to talk about it. But the idea is that we really want to try to look at the cases where we've got cycles that share intermediates between two cycles that deliver a product from one cycle that might be a very activated species into a second cycle. How can we use kinetics to understand these types of cycles the way we've been doing with with standard simple cycles? We think we have this down, and then we also eventually want to bring in deactivation. We want to understand what happens when radicals fizzle out somewhere else, or we have side reactions. But I think we have to get started on thinking about what kinetic profiles can look like in these more complex cases. And I'm actually I only came here from Liverpool, not from Southern California. I'm on sabbatical when Andy Cooper's on it, and working with AI-Chemie with Imperial. We're actually trying to develop ways to look at the workflows to do automation. To get automation doesn't have to mean 1,000 different ligands with one data point each. It can be 10 ligands and 100 data points of 10 and 100 data points of time course. Data density doesn't have to be one piece of information from each reaction. That's my main soapbox, is when people are talking about automation and high data density. We can use these robots, take samples, take it. If you've got a 96-well plate, use 48 of them for your reaction, and then the other 48 can take the robot and make a quick quench. So then you can send those samples to NMR and spec or HPLC, and use the form of the kinetic profile to try to help you understand what's going on in the reaction. So I think that's it. Actually, this is from back home. We actually do get rain in San Diego, maybe 3 or 4 times a year. It rains. It's not like 3 or 4 times a week in Liverpool. But and this was one day when this is actually where my office is. Well, it was. I stepped down as department chair, but it was here. It goes across the parking lot to the Flame of Knowledge sculpture, which I thought was really fun. But actually, I want to say that this work, the ideas in this really started when I was at Merck. But it actually came from a project that UPSRC funded in the early 2000s when I was at University of Hull. It was called For Microgrids and Multikilos. It was Asteria Stavridis at UC London, me, and Stan Roberts at Liverpool. And we had 13 companies, and we were basically trying to look at organic synthesis all the way through to production, from pharmaceuticals. And so that's really where this technology or this methodology came out. Leverhulme Trust is paying for my sabbatical. I'm working, hopefully, they pay for me to have some salary, but no people. So Becky has been very nice to work with me with students in Imperial, and then Andy and Jeff Ames in Liverpool. And so I've been involved in a couple of different NSF centers where these methodologies have been used. And I had a grant to look at temperature scanning reactions. Maybe I would have had time to talk about it, but we developed a method where we actually scan temperature at the same time. You know, RPK is based on having a concentration scan. You watch concentrations change from the beginning through to the end. It's like an initial rate data every 2 seconds. And I'm thinking, what other variables could we make continuous? We've taken an initial rate measure, which is discrete, and made it continuous. What else could we do? Well, you can't really make the catalyst continuous, and you could put it, syringe it in slowly. That probably doesn't make much sense. You can't make additives continuous. There's a lot of things that have to be discrete experiments, and we usually do discrete experiments for temperature. You know, we do isothermal temperature 1, temperature 2, temperature 3. We build an RNA plot. We can actually do that all on the fly. We have a project going on now with the collaboration with Phil Barron. He just had 2 papers on nickel catalyzed. One was stereo-retentive coupling, and we're looking at the non-stereochemistry system. But from doing this temperature scan, we scan temperature at a constant ramp while the reaction is going on, and we apply RPKA and get iron plots and reaction orders all out of the same stuff. And that was funded by this grant here. And so we're looking for problems to look at. I'm a chemical engineer. You know, the molecules I just said, I only learned from my organic chemist colleagues. So, you know, we really want to look at real life, really interesting, important problems and see if this kind of, if my blind faith that looking inside the reaction will actually help us understand it better. If that holds true for complex systems, I want to look at systems where we don't already know the answer. You know, I want to look at systems where we can apply this right from the start. So email me either I'm on the website at Scripps and at Liverpool, and I really want to set up collaborations this year. So with that, I just want to have one last slide, and that is. I like to mention John Martin. You may have seen on the first slide that I'm the John Martin endowed chair. He was a visionary. He was an amazing scientist. He ran Gilead, the CEO and president for years. He retired. He was actually just set to become the chair of our board, our advisory board at Scripps, our board of trustees at Scripps. He tragically died very suddenly. He's responsible for a lot of the AIDS drugs, HCV vaccine. You know, and one of his degrees is chemical engineering. So he's an organic chemist and a chemical engineer. He was. And so it was a really lovely thing. He and his partner gave the money for the chair personally, because he said, "Oh, Gilead never gave you guys money," but they supported the chair. So I just think it's really important to remember people like this, because he was a visionary basic scientist on the industrial side, solving real problems. So I think it's a nice thing to make sure people don't forget. So thank you very much. I'd be glad to answer your questions. Yeah, Kenny, and that's a great lecture. Okay.
Thanks, Donna. You mentioned something interesting at the end, and that is catalyst deactivation. So real catalysts are plagued by deactivation of some form or another, and the initial rate analysis obviously conveniently ignores those, right? But with RPKA, it's always in the data. So I'm curious to hear your thoughts on how we should spot and analyze and understand deactivation in reaction profiles.
Yeah, I mean, RPKA does that. It's the same access. I didn't have time to go into it, but anyway, same access protocol does 2 different reactions. When you start, let's say you're starting with simple numbers, 1 molar of substrate A and 1.2 molar of substrate B. At some point in that reaction, you get down to 0.5 and 0.7. Because you use one of each. Why don't we start another reaction at 0.5 and 0.7? a different vial, not the same vial, to start it, and then look at those two kinetic profiles. If they overlap, if they're going at the same rate, that tells you the catalyst isn't deactivating and the product is not inhibited. That reaction that I showed you of Jim Kwan Yu, where we had negative order, they were inhibited. The product, and so we did that. We did same excess, and we saw that they didn't overlap. We added the amount of product that would have been in that same, and then they were. So, and I think the other thing is Jordy Burroughs' VTNA for catalyst order. If you combine that with same excess, you have really nailed what that catalyst is. From the beginning to the end, reaction. There are some systems, if you've got a lot of the same plated catalyzed head coupling reactions where you make dimers of the catalyst, that will change. When you're trying to look at catalyst order, you change the concentration of the catalyst. You might change the amount of dimer versus not. That's why you get weird orders, like not all 0.5 and not 1. And so those two methods together, VTNA and I don't know if you know what that is, but it's an amazing. It was a hot paper in Underbudge about 10 years ago. And I went to give a seminar at work the week after it came out. I'm like, you guys have got to look at this. We're on it. We already saw it. We're doing it every day. It's a really fast way of looking at catalyst order. So between those two, I think, so that's what I said to Jim Kwan with this system. Okay, I've shown you it's deactivated. Now back to you, organic chemists. You figure out what to do about it. So it doesn't tell you what to do about it. It tells you what to say. And I think the same thing in so I didn't show you in those simulations. They have simulations, so the catalyst order was one. But if we have deactivation, then we have to account for that in the kinetic models. And if you're going to do Dynacam or something like that, you just or capacity, which is then you put in some kind of deactivation step. That you still start with the orders, and then if you have, and then you can go, then you have to figure out, what can I do? How can I get this product off the catalyst if it's deactivated?
Yes. Thanks, Donna. Parimala from the University of Nottingham. So increasingly in the UK, we're looking at using biomass to make bio-based chemicals. So, and the biggest challenge is where we're getting our biomass from. So to create drop-in replacements. For example, I collect biomass from Lincolnshire, which is very different to what it is in Wales. And we're trying to do exactly what you've shown, but using enzymes, and we still linearize to a certain extent because we're trying to understand what's happening. Do you have any advice on almost like feedstock agnostic modeling for multi-component when you have seasonal and geographical variations in biomass when you're trying to create drop-in replacements for chemicals?
So first, if you have what you would call a standard feedstock, you're starting from and you've developed, say, an RPK model for how you think what's binding and how it's reacting. It's much more complicated than what I'm talking about. But then what I would do would be, I don't actually care about rate constants, numbers. I don't care. I care about the profile. And we can see in profiles. So if you see a fast rate and then there's a curvature, like a hockey stick, and then it goes down, that's got to be something like catalytic, a mechanism change. And people try to hand wave into telling me I'm an editor, associate editor, Journal of Organic Chemistry, and they try to tell me all sorts of things that. But so I would check. First thing I do with the new feedstock is say, does this profile doesn't have to be the same rate? It's just the same sort of. Is there anything weird about the profile? Does it look similar? And you can see, like, it might change the resting state in your enzyme system. That's easy, like I showed you in the slide. I didn't put it up again. Maybe way back. Sorry, but you can see from the shape of the curve, something about where you get some. If you do it for 40 years, you can. I'm almost as good as they are, But but this is actually what we want AI to do. That's what I'm trying to do with this sabbatical, to say, look at the form of the curve and see if it's, and that alerts you. Okay, this one's different. Am I getting a low yield? And you can do high throughput screening just to get full to the profile. Andy said something to me when we were decided to write this proposal. I'm like, I have to figure out. We have got all these MLAI methods like Met Sigmund. They do great work, but they use these descriptors, okay? And they come up with, they do 100 reactions and as tests and then they do a validation, a bunch of reactions, and then they get a prediction. So they do like 140 reactions to get 4 data points. Yay, we predicted it. We're hoping that we can do it with fewer data points. Plus, the thing that always bothers me about that is that you basically get, in those plots, you'll have delta delta G observed versus delta delta G predict. One piece of information from each of those reactions. On that plot, we don't even know the yield. We have known nothing about the kinetic profile. They use bond angles, steerable parameters, varied volume, all these descriptors. And to me, it's offensive. Sorry, I love math, really good friends. But my whole life has been based on saying, looking inside the reactions is important. And if you don't need to know anything about the inside, and only the EE at the end, then I see my life pass before my eyes. So I think looking at so what Andy said when we were writing this, he said, look, if you're on the air, they look at your eyes and they notice you. They notice me and not you, right? Do you think these programs could discern differences in kinetic? We haven't figured out exactly how we're going to do that yet. Figure it out. But I just believe that there's information in there about the mechanism. It may not be one. It may boil it down to say 5 instead of 50 mechanisms. That's still better than not looking at it. So form of the kinetic profile. That's and actually you got to do 2 of them because some of these forms look just the same. If you do different excess, you can wait. I've got negative order in my substrate. How can it be? So do the 2 standard reaction, same excess, different excess. 3 reactions for every 3 vials, and then take 10 samples out of each vial, and then do that for however many ligands. And we all want to say we've got 1,000 different data points. If we get 1 data point for each of 1,000 different phosphine ligands, do we know much about those reactions? I would bet that we could take any of those things that didn't really look like hits and optimize them into a hit. So more information on a fewer trial possibilities, I think will get us further faster.
You about electrochemistry and the fact that systems are not stirred and how you deal with diffusion information.
It can be a really big problem because the bilayer right at the electrode can be difficult. Some of the systems that we've looked at, we haven't looked deeply, but I'm part of this NSF center where you use mediators. So mediator does stuff at the electrode, and then all the catalysis is out here. Typically, that you can lump that diffusion into a rate constant for the mediator cycle. So I haven't really dealt with. I think Klaus Helgaard and Mimi have done some stuff on looking at that, and that will be an issue. But you'll know it right away. If you get 0 order kinetics, then you say, okay, why? Is it because of the reasons I showed that are kinetic reasons, or is it a mixing reason? There are many cases in the literature of 0 order kinetics in hydrogenations that they say, oh, it's strong binding with substrate, where it's actually the slow step is getting hydrogen from the acid. And we know how to deal with that issue. That's easy. But right we did a couple of papers in collaboration with Phil Barron, where we pumped from the electrochemical cell through a transmission IR and back to this recycle flow. And we were able to get really good kinetics and reasonable rate law made sense. So in that particular case, it looked like diffusion wasn't an issue. But I think it's going to be case by case.
Yeah, related to that, in your methodology, it looks like you make estimates of the equilibrium constants.
Yeah.
By various powers, right?
Yeah, exactly.
And so the question is, you say the minimum number of experiments is 3, but what happens if you do 300, and how much better does the refinement on the?
I don't think it's
on the number.
Basically, when I do 3, I look to see if same different standard and different excess overlay. If they overlay, I don't need 300 more overlay.
But you could do other excesses. You could do
Other excesses. We usually do these. So mathematically, the system, if you have 2 unknowns, you need it's algebra, right? Remember simultaneous equations? You need 2 independent equations to solve for 2 unknowns. Different excess gives you 2 standard. Do a different excess. That's an independent reaction. So it's your 2 equations. Same excess is not independent because it's the same reaction, just started from a different place. So you need same excess to see if the catalyst is getting tired out, or the product is inhibiting. Different excess, it's mathematically defined by those two. We usually do at least standard and then 2 different excess on top. But we're talking about 4 or 5 or 6 in a new product inhibition. In a half dozen experiments, you can have a really good idea of where to go next, which is much more than 1,000 data points of initial rates.
I'm absolutely convinced.
Thank you. I did a good job. Speaking to the choir, yeah.
Hi, Donna. Your lecture took me back, and one statement in particular, when you said, you can use the kinetics to prove or disprove the mechanism, it took my PhD supervisor 2 years to forgive me for destroying his mechanism. And I think you'll remember that work.
They're very wedded to their.
They do.
It can be very, very tough. But
The good old days of alpha keto ester hydrogenation.
yes. Oh, yes.
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 We've been doing some work recently in my previous employment, developing a kinetic model so that we could scale up chemistry, so that we do the minimum number of experiments in the pilot plant before going into the demonstration plant. Do you have any examples of how, based on the work you've done with partners, you've accelerated the scale up of any chemistry?
So in consulting, I have, but usually it goes from what I did, and then they go on and we'll probably be able to talk about it anyway. But yeah, in fact, the first thing when I left Merck and I was in Germany, went to the Max Planck Institute, I got an email from Ed Grabowski, who's like God. That's this entire God. He was God of process chemistry. And he wrote, he said, we just did the pilot run and the stuff he did for us. And, everything went smooth. Nobody had to get up in the middle of the night and base it. And it was wonderful. But, so, yes.
So codifying into Aspen Plus and models like that? Codifying into Aspen Plus for the real big scale
I
ups?
haven't. So I've done a little bit of work with people using Dynachem where they put in massive heat transfer and other things that I'm not considering. I'm just looking at if I used to say this about hydrogenations. If you do it correctly, it's scale transparent. It should not, no reaction should change with scale. If you understand the physical rate processes, as well as you understand the chemical rate processes, your goal is to make the chemical rate processes dominant in any system. How you do that? You need good engineers as well as chemists. But so I haven't, like I said, Dynachem is the one system that I've ever worked with. Right now, there's a trend. There's been a couple of papers out where people in pharma companies have been trying to sort of ML/AI approach. Actually, Joey Burris had a really nice paper in Nature a couple of years ago showing like how you could use neural networks to identify reaction mechanisms. And so people sort of go to town on that. They come up with like 50, there's one group I'm consulting with, they had like 56 different mechanisms. Look, and we picked the right ones. You didn't need 56, really there were about 4 or 5 that you could choose from. And so we kind of set up strawman when we look at 1,000 different ligands, or we look at 1,000 different reaction mechanisms and pull one out because it makes us feel. I think we should spend more time on, basically looking at fewer variables, understanding it, and then go out to. If you understand the mechanism, then you try another phosphine ligand later. Oh, it's a different profile. Must be a different mechanism, or must be a different rate length. You're not going to get that from 1,000 data points of 1 point yields. So I haven't. The answer to your question is, as a chemical engineer, I'm embarrassed to admit I've not done any scale in more than 30 years. But I've been involved in teams in consulting that have, and this is the first step. They're all doing it. When I first published this, it was really hard to get published. People thought I was a heretic. She's doing. They thought it was cutting corners. I think this is actually more rigorous than initial rate kinetics because you're sticking near the conditions that you actually do the reaction. So 10 equivalents, which they can't do in pharma. They started using it right away. At first, when I saw papers in RPA plots, was either me, students who had been trained in my group, or pharma people. And so now other people are grudgingly. There's a few big shots. I won't name them. Maybe in the bar tonight, I'll tell you, but who still think I'm heretic. And I have a slide. I don't think I have it in this stuff, but I showed when I gave talks in pharma about the review comments because they're just amazing. And one of them that said, "Okay, I get it. Your method is liable to be okay, but it's liable to be used by non-kineticists." the point, you know? Like, you don't want to learn how to do Hamiltonians to do your NMR. Somebody this morning, another talk was saying, all this has been worked out for you. So it's actually automated to that extent. You do Fourier transform IR without doing Fourier transforms. Why can't you do kinetics? Like the mechanistic method. You know, once somebody has shown, maybe you should go through it once to understand how it's done. But once you believe that, then you don't have to be a kineticist to get, you don't have to be an NMR specialist to get good NMR data. And then the second part of the sentence after that was, those drug companies. So I love giving that when I give a seminar on farming. So I've been on both sides, so I'm allowed to say that. But I mean, why would some academic think that he's so much better than people at farming companies? They actually have to make it. They can't just stand up and give a talk and have a clap. Well, that's nice. But it's weird.
I wish that I think we'll stop and then I'm in.
Oh, then you- Did I go?
Thank you very much. Yeah.